In this lecture, we'll cover Chapter 16, the nonspecific defenses of the host. The nonspecific defenses of the host are those defenses which are not particularly part of the immune system, which we will be studying in Chapter 17. Before we begin a discussion of anything concerning host defenses, it's important to make sure we have a couple of terms straight in our minds. The first of those is actually the non-specific defense of the host. And sometimes, depending on the textbook, this will be called the innate defenses of the host. And whether you're talking about non-specific or innate resistance, you're talking about a generalized defense against any and all pathogens. So really either of these terms describes what we're talking about, just in different ways. Non-specific resistance describes the aspect of this resistance which is targeted at any pathogen that might come into contact with the body. The innate resistance highlights the fact that these are defenses that we are born with, fully functional, fully capable, and ready to defend against any pathogen. Those defenses, which we will cover in Chapter 16, are contrasted with the immune system, which is a specific adaptive response to a particular pathogen. So in Chapter 17, we're going to be talking about the specific adaptive resistance of the body to particular pathogens. And when we talk about that, we're actually talking about the immune system. So these defenses are sort of set up in a way such that the pathogen first encounters the nonspecific and innate defenses, and then if those defenses are combated and beaten, then that pathogen will be exposed to the specific adaptive resistance of the immune system. So you see here in the diagram, you have some kind of pathogen, maybe a virus, a bacterium, fungal cell, protozoan, you name it. And the first thing that aims to stop those things is intact skin. We're going to find out as we talk about body defenses that intact skin, intact mucous membranes are actually our first and best defense against microbial infection, which remember is the colonization of the host by pathogens. So intact skin, intact mucous membranes are great defenses. In addition, on the skin and mucous membranes, we have different antimicrobial substances there. Some of them are made by our normal flora, some of them are made by us. Even just sweat itself is antimicrobial. And if those are breached, if the skin is torn or broken, or there's been some damage to the skin, or if the mucous membrane is damaged, then the pathogen is able to move through that first defense. And even there, it's still going to be interacting with non-specific and innate defenses, which we're going to be talking about in this chapter. And those specifically are the process of inflammation, the process of fever development, and a cellular process called phagocytosis, which is accomplished by particular white blood cells, which are grouped together and called phagocytes. Now, if all of these defenses are engaged, and still we have microorganisms colonizing the body, then the immune system comes on board. And we're going to find in Chapter 17 that the immune system actually is further divided into what are called the humoral and cellular or cell-mediated branches of the immune system. Another couple of terms that we want to keep in mind as we talk about host defenses is susceptible and resistance, which we did talk about in Chapter 14. Remember, susceptible individuals can contract a disease because they are not resistant to that disease, whereas resistant individuals will not contract a disease because their defenses are able to ward off the disease prior to any disease syndrome being established. Now, in your lecture notes, if you're in the habit of printing them out, you will find a summary table of the nonspecific and innate defenses, which we're going to be going over in Chapter 16. As I mentioned previously, your first and best defense is your physical barriers and your normal flora. So we have here a diagram of the body. And obviously the internal organs are shown on the outside. But if we kind of go clockwise around that, we can start up in the upper right-hand side and we see the respiratory tract. Some of your physical barriers that are helpful there to keep you from being colonized by microorganisms would include lots of different mechanisms that are meant to move air and microorganisms and mucus out of the body, and that would include coughing, sneezing, nasal secretions, which you would then blow from your nose. The hairs and the ciliary escalator hairs in the nose are meant to trap microorganisms. Ciliary escalator is a really interesting organ that is found in the respiratory tract, in the lower respiratory tract, and it beats upward. So as things are trapped in the mucus of the respiratory tract, rather than being able to slide down into the lungs and establish a lower respiratory tract infection, the ciliary escalator will move those things up into the upper respiratory tract, where a person might clear their throat and move that mucus with trapped microorganisms out of the respiratory tract. In addition to that, we have something called fixed macrophages, which we haven't discussed at all to this point, but macrophages are a type of that phagocytic cell that we mentioned earlier. And in parts of the body where interaction with pathogens is very likely, you'll typically find macrophages stationed in that area, sort of as a sentry for the immune system. And we have lots of fixed macrophages in the respiratory tract, in the lungs, in order to combat any would-be pathogens there. They're also found associated with the gastrointestinal tract, and the reason for that, as we're going to find out later on, is that the respiratory tract and the gastrointestinal tract are the most common sites of infection, with respiratory tract actually beating the gastrointestinal tract out for first place. The respiratory tract is the most common, what is called portal of entry for microorganisms, and then the gastrointestinal tract comes in at second place there. And because of the fact that so many pathogens want to get into the body and establish infection through the respiratory and gastrointestinal tract, we see lots of fixed macrophages there, kind of just as sentries waiting and watching in case that happens. So that brings us to the gastrointestinal tract, which of course begins at the mouth, ends with the anus, and is a mucous membrane lined tract where lots of different things come into play to protect you, especially because you are intentionally putting things from the outside environment into this gastrointestinal tract when you eat food. So right off the bat, when you eat food and you start to chew it, your saliva contains an enzyme called lysozyme, and lysozyme is an enzyme which lyses bacterial cells. So even as you're chewing your food, you're sort of disinfecting your food. In addition to that, as it moves from the esophagus into the stomach, you've got a very acidic stomach there, right? A pH of around 2, which will actually kill pretty much all microorganisms that are not overwhelmingly populating the stomach. And once that food moves out of the stomach into the intestines, there's a lot of competition by normal flora that will sort of crowd out would-be pathogens. And your gastrointestinal tract also has a way of moving things out of the body. So when you vomit or you have diarrhea, a lot of times that's a result of your body registering that there is something in your food that ought not be there, and so your body increases peristalsis, and if it increases peristalsis to move things down through the gastrointestinal tract, you will get diarrhea because your body hasn't had enough time to reabsorb water from that food. And if you still have food in the stomach, your body will begin a process of very violent peristalsis, which results in you expelling the food through your mouth in what, of course, you know is called vomiting. So gastrointestinal tract, like the respiratory tract, has that ability to move things out of the body, either up and out of the body as the respiratory tract does, or down and out of the body through the lower end of the gastrointestinal tract. Other body systems that come into contact with the outside world include the urogenital tract. Urogenital tract, especially in females whose urogenital tract is much more open to the environment, have a tremendous amount of normal flora whose presence there protects and keeps that environment healthy, keeps it from being established by pathogens. In addition to that, if anything, including the normal flora, were to move out of the exterior urogenital area and move into, say, the urethra, urine actually serves a dual purpose of removing metabolic waste from the body and rinsing the urethra. So again, you see this movement out of the body that counteracts any microorganisms trying to move up into the urethra and the bladder in order to establish cystitis, which is the technical name for a bladder infection. In addition to that, urine is typically acidic, which, as you know, makes it difficult for mesophilic organisms to survive, let alone grow. And so each of these body systems, which are open to the external environment, have these innate defenses which have been there since the moment you were born, right? You've all been producing urine since before you were born. You are colonized with normal flora the moment you're born, and all of these other things come into play as well. In addition to that, as we mentioned, the skin is a highly beneficial structural barrier to most microorganisms. So intact skin, as I mentioned before, is going to be a very important barrier for you. In addition to being a physical barrier, lots of acids are produced by glands in the skin that inhibit the growth of microorganisms. And again, you've got a tremendous amount of normal flora there that will crowd out would-be pathogens. The last place that we're going to discuss that is open to the external environment is the eye. The eye is a particularly dangerous place to have colonized by microorganisms because the optic nerve allows those microorganisms sort of a straight shot right into the brain, because really the eye is kind of an outcropping of the brain itself. So the eye has lots of different defenses. And one of the things we're going to learn later on in chapter 17 is that the eye is a place where the immune system can't actually function in the way that it normally would, because the normal functioning of the immune system causes coagulation and clumping. And if you have that process happen inside the space of the eye, you'll actually cause blindness. So the eye has a lot of innate defenses that are meant to protect it. One of those is blinking, right? If you've ever gotten something in your eye, you notice it makes you blink repeatedly. And if you've ever felt like you have something in your eye, maybe you have intentionally blinked. And as you blink, you actually close your eye and allow tears to flow from the upper outside corner of your eye across your eye, washing it. And then those tears are actually collected in the inside lower corner of your eye, and then those tears actually drain through the nasolacrimal duct into your nose, which is why if you cry, you oftentimes get a runny nose, because those tears are flowing out of the eye and into the nose. And that's a really important process because it takes all of those microorganisms that are trying to establish an infection in the eye, moves them out of the eye into the nose, where either a runny nose will run them out of the body, or blowing a runny nose will remove them from the body. And while they're there, those tears are killing the bacteria, because the tears, like saliva, contain lysozyme, which is digesting bacterial cells, as those cells are bathing in the tears. So that's our first defense, our physical barriers and normal flora. We're going to move on now to sort of a second round of defenses that are still considered innate defenses. They're considered a second round because they come on board if these first defenses, the physical barriers, are breached. And the first of those is actually a response to the damage of skin And that is called inflammation. So I know inflammation is a word that people use sort of colloquially. Something's inflamed, it's swollen, but inflammation actually is a process that happens in response to cellular or tissue injury and then results in these 4 very important signs and symptoms as a result of tissue injury. So this second defense comes on board not in response to bacteria or any other pathogen, but in response to cells being damaged. So on the left-hand side of this picture, you see a pin stabbing into skin. Now, I don't want you to pay attention to these bacteria here because this could also be a sterile pin that's stabbing the skin, and the result would be exactly the same. That pin damages skin cells, and the skin cells, when they get damaged, release chemicals, and those chemicals tell the body that the first defense has been breached, right? And if the first defense has been breached, the body's natural assumption is that microorganisms are going to come into the body. So that sort of sounds an alarm for the body. And those chemicals cause a couple of things to happen. Initially, those chemicals cause the capillaries in the area to dilate. So you notice if you look at these 2 pictures, you look at the one on the far left and the one in the middle, these pink lines that are supposed to be the endothelial wall of the capillary, notice there's space between them now. And that space is really important because it makes it easier for phagocytes, which we're going to talk about in a few minutes, to actually move out of the blood vessels and into the tissue space, right? So they can get in there and see if there's anybody that they need to worry about. So when those capillaries start to leak, the white blood cells can come into that local area, that tissue area, and that process of moving out of the capillary and into this tissue space, depending on the book you're reading, will either be called extravasation of the white blood cell or phagocyte, or it'll be called diapedesis of the white blood cell. But you notice in these 3 pictures, right, you see the bacteria coming in on the pin, the capillaries become leaky, they move out of the capillary and into the tissue space, and then they phagocytize those bacteria. So when that process happens, other things happen as a result of it, right? So capillaries become leaky. The other thing that will increase the movement of phagocytes into the area is that local arteries will start to dilate. So the circumference of the local arteries will get larger because the arteries relax, and that causes increased blood flow into the area. And that increased blood flow into the area carries with it white blood cells that are just traveling through the bloodstream, sort of monitoring what's going on in the body. So you'll notice in that middle picture, the arteries dilate and the capillaries leak in response to this chemical. So that chemical causes both of those things to happen. Now, how do we see that process occur? What do we notice as a sign or a symptom of this process? We notice 4 things, and those 4 things are listed for you across the top of this picture. You might not recognize those 4 things because they're actually listed in Latin. And the 4 responses or signs and symptoms of inflammation are calor, rubor, tumor, and dolor. And those of you who speak Spanish or another Romance language are actually going to have a leg up on the rest of us because you might recognize calor and dolor especially as words that mean redness and pain. So calor is a Latin word for redness, or excuse me, heat. Rubor is the Latin word for redness. Dolor is the Latin word for pain, and tumor is the Latin word for swelling. So the 4 signs and symptoms of inflammation are calor, rubor, tumor, and dolor. So why do those things happen? Well, calor and rubor happen because of those arteries dilating. So when the arteries dilate, right, the inside of the artery gets bigger, the circumference of the artery gets bigger, and blood flows into the area. And what color is blood? Blood is red, which gives us that first sign right? Because it's measurable and you can see it, of inflammation, rubor. So redness is caused by the arteries dilated and increased blood flow to the area. Now, in addition to being red, blood is also hot because it's coming from the core of the body. So when those blood vessels dilate and the warm blood comes into the area, not only does it make that area red, it makes that area warmer. So the arteries dilating and bringing increased blood flow to the area are responsible for two signs of inflammation: calor, which is heat, and rubor, which is redness. Now, the leaky capillaries that we discussed previously are responsible for the last two signs and symptoms of inflammation. And that happens because as those capillaries become leaky, in addition to white blood cells being able to get out of the capillary and into, excuse me if I said red blood cells, I meant to say white blood cells. White blood cells getting out of the capillary and into the tissue space, the fluid of the blood can get out of the capillary and into the tissue space. Now, it's important to notice that fluid, but not red blood cells, are capable of moving from the capillary into the tissue space. So that fluid is not responsible for causing redness. That fluid in the area causes edema or swelling, and you'll remember that tumor is the Latin word for swelling. And tumor, again, is a sign because it's a measurable, visible result of inflammation. The fourth and final result of the inflammatory response is also caused by capillaries leaking, but it is not a sign, it is a symptom, and that symptom is pain. Increased fluid in that area causes pain, and that pain is registered by the person, right? It can't be measured by someone else, but someone says, yes, it's painful, right? So you see that it's hot, it's red, it's swollen, and they articulate to you that it's painful. And all of those things together tell you that there's an inflammatory response going on in that area, right? Because there could be something coming in with that pin that's foreign and pathogenic, and so your body brings those white blood cells into the area very quickly in order to deal with whatever threat might come with that tissue injury. So those white blood cells that we're talking about moving into the local area are just one kind of white blood cell, and they're the only kind of white blood cells that we're going to be talking about in chapter 16. So we have a picture here with lots of different kinds of white blood cells and red blood cells and other blood components, all of which come from a particular kind of stem cell, which is a pluripotent stem cell called a hematopoietic stem cell, which means it can become blood cells. So those stem cells in the bone marrow and things like that are constantly being produced and differentiating into what are called myeloid stem cells and lymphoid stem cells. Notice we don't really talk about the myeloid stem cells here at all, but those become your granular leukocytes, your platelets, and your red blood cells. On the other hand, you have this lymphoid stem cell, which differentiates into monocytes, T cells, B cells, and natural killer cells. Now we're going to come back to T cells and B cells in chapter 17. Today, we're just going to be talking about that monocyte because that's the precursor to the phagocytic cells that are working really hard within the innate defenses in order to combat infection. Now there are other cells, right? You might have learned about neutrophils. Neutrophils are also really important in combating infection, but the reason we're paying attention not to the neutrophils but to the monocytes is because the monocytes, we're going to find, are really important because they communicate between the innate defenses and the immune system. So not only are they out there, right, moving into the tissue space because of inflammation, but they're also reporting back to the immune system and letting them know what they find. So they're not just neutralizing threats, they're actually bringing the immune system into the fight in order to fight well, fight hard, and recover from infection. So we're going to be talking about dendritic cells and macrophages today and the work that they do to phagocytize antigen and also the work that they do in order to communicate with the immune system. So macrophages and dendritic cells are phagocytic cells, which means they are capable of a process called phagocytosis, which you can see pictured here. Phagocytosis begins with chemotaxis, right? Actual movement, motility of the phagocyte toward the microorganism. So it will actually register chemical signals in the environment, and it will move toward that microorganism, and it will adhere to the microorganism. Adherence of the phagocyte to the microorganism causes the phagocyte to engulf the microorganism, right, and to ingest it into a vesicle called a phagosome, which literally means eating body. That phagosome contains the microorganism that the phagocyte wants to eat and to digest, and so the phagocyte will fuse that phagosome in step 4 with a resident lysosome. And that resident lysosome will fuse with the phagosome, and it will become a phagolysosome. And within that phagolysosome, in step 5 there, you see those lytic enzymes and digestive enzymes of the lysozyme digesting that microorganism and neutralizing the threat of the microorganism. And then once that microorganism has been completely digested, the waste material from that microorganism is discharged into the space, the interstitial space of the tissue. So how is it that microorganisms are recognized by phagocytes as something to attack? Well, they're recognized because of a very special kind of a receptor on phagocytes, which are called pattern recognition receptors. So pattern recognition receptors, which are abbreviated PRRs, are found on phagocytic cells, and the patterns that they recognize are pathogen patterns. So pattern recognition receptors are found on phagocytes, and they recognize pathogenic patterns. So when they recognize that pattern, they recognize that the thing that's attached to it is a pathogen, and that will cause them to engulf whatever it is that they've attached to. Now, I've added the name of one particular kind of pattern recognition receptor here because it's one that your book uses as an example. And it's actually the one that's given as an example here, and that is the toll-like receptor. So the toll-like receptor, which is pictured in the inset of this diagram here, is a pattern recognition receptor, which is on the phagocyte and is capable of binding a pathogen pattern. The thing that that pattern recognition receptor binds is called a pathogen-associated molecular pattern, or PAMP. So PAMP, as you can see here on the bacterial cell, is bound by the toll-like receptor, and in this case, the pathogen-associated molecular pattern is peptidoglycan. So the bacterial cell's peptidoglycan is actually recognized by the phagocyte as foreign. Flagellin, which is the protein in bacterial flagella, the lipopolysaccharide outer membrane polysaccharides are recognized, viral nucleic acids are recognized, lipoteichoic acid, which is found in bacterial cell walls, are all recognized, and they all group together as these pathogen-associated molecular patterns, or PAMPs. So you got to keep straight, the PRR is on the phagocyte, the PAMP is on the pathogen. So the pattern recognition receptor recognizes the pathogen-associated molecular pattern. And when that happens, something in addition to phagocytosis actually happens. And these activated phagocytes, once their PRR has bound PAMP and this process of phagocytosis is occurring, it actually causes that phagocyte to become activated and to release chemicals which will increase the other innate responses. It will increase inflammation. It can actually turn on fever, which we're going to see in a couple of minutes, and it can interact with complement proteins in order to recruit more innate defenses, right? Because if one bacterial cell was found and digested, chances are there's millions more where that came from, and so it's not going to be adequate for this one white blood cell to cruise around and try to find all of the pathogenic cells. So this is where you see these guys interacting directly with pathogen, acting as an innate defense. But I mentioned that in addition to that, monocytes also communicate with the immune system. So these monocytes, which differentiate into macrophages and dendritic cells, function as scouts who reside in the tissue space, move through the the cardiovascular system, and are constantly interacting with all of the parts of the body in order to see if anything's out of whack that needs to be communicated to the immune system, because the immune system cells stay in the lymph nodes. So they're kind of up in their ivory tower in the lymph nodes, don't really know what's going on unless the monocytes, the macrophages and dendritic cells, have brought something to them and let them know that that thing is bad. Now, a very black and white understanding of macrophages and dendritic cells begins with a very black and white thought about what they look like. So we have two pictures here. The upper picture is actually a macrophage, and that is like a beautiful textbook, amazing example of a macrophage. The problem with that picture is that macrophages do not have a cell wall because they are human cells, and so they have no fixed shape, which means that if you took that picture 3 seconds later, the shape of that cell would be very, very different. But the very stereotypical shape of a macrophage is to have this very large body and these teeny tiny hair-like projections. And on those hair-like projections is where you have those PRRs that are looking to bind with PAMP. Now on the bottom here, we see a very classic textbook example of dendritic cells. And those of you who have taken physiology and have seen what dendrites look like, you might be thinking to yourself, I get it. That's why they're called dendritic cells, because they have these long branching projections that resemble the dendrites of neurons. But they are not at all like neurons. In fact, those long branching projections are areas where there's a high concentration of PRRs, pattern recognition receptors, looking to connect with the pathogen-associated molecular patterns of pathogens. So those are very textbook, but again, we don't have cell walls, we don't have a rigid shape, so the shapes of these things are a lot of times a lot more difficult to pinpoint. And the good thing about that is they both do the same thing, right? When their PRR become activated, they phagocytize the pathogens. They recruit the innate defenses by releasing chemicals that sort of sound a chemical alarm. And then they very importantly take that thing that they've phagocytized and they present it as antigen to the immune system. So macrophages are typically found in the tissue and they present pathogen to B cells. Dendritic cells typically migrate to the lymph nodes where they present pathogen to T cells. Now that all sounds well and good. The only problem is B cells are found in the lymph nodes. So a macrophage can't stay in the tissue and also present pathogen to B cells, but the way that these things are discussed and the way that these guys are differentiated from one another is in textbooks. You'll find macrophages stay in the tissue and present pathogen to B cell. Dendritic cells migrate to the lymph nodes and present pathogen to the T cell. For our purposes, what we really want to focus on is what do these cells do, right? They phagocytize pathogens, they recruit innate defenses, and they recruit the immune system, right? Very, very important because they're directly defensive and they engage both aspects of the body's defenses, both the innate and the immune system defenses. So we're going to hear lots about these macrophages and dendritic cells as we move through and talk about the body defenses. The second innate defense, once you've gotten through the skin, is fever, and fever is actually caused by white blood cells, macrophages, and dendritic cells communicating with the rest of the body. Now, fever, it's important to understand, is not hyperthermia. Fever is not, ooh, I'm hot and I'm going to start sweating and vasodilating and do all these things to try to cool myself down. Fever is a resetting of the body's thermostat. So these chemicals move, as you can see, from a macrophage or dendritic cell that's become activated and phagocytized pathogen. When it phagocytizes the pathogen, it releases these chemicals, which move through the blood vessels, through the blood-brain barrier, move up into the brain, get to the hypothalamus, and cause the hypothalamus to produce a chemical called prostaglandin, which resets the body's thermostat. So no longer does the body's thermostat say we want to keep everything at 98.6 degrees. Now the thermostat says we want to ramp the body's temperature up to 100 degrees. And that's why when a person has a fever, right, you'll see them shivering or doing other things even though they're already hot, right, which is different than if a person's just out on a hot day, right? A person's going to fan themselves and sweat to try to cool down. But a person whose body temperature is 99 and who has this defense going on inside them, their body's actually trying to ramp the temperature up higher. So they're going to shiver, they're going to vasoconstrict and do all of those things that you would normally do when it's cold outside. So why does the body do that to you, right? Well, there are a couple of different things that are advantages of having a little bit higher body temperature, right? A low-grade fever. So 101, 102, right? Anything above that, not super helpful. But the advantages to having a low-grade fever is that it increases the level of something called transferins, which scavenges iron. And it's really important to scavenge iron because lots of viruses and lots of microorganisms need iron, right? If there's low iron availability, it inhibits their growth, it inhibits their RNA and DNA synthesis, right? It can change their cell morphology and make them easier to phagocytize, so bad news, right? In addition, a fever will increase interleukin-1 activity, which we're not going to get into, but just sort of recruits other body defenses. It also turns on something called interferon, which we're going to talk about in a few minutes, and it speeds tissue repair, right? Because a little bit of increase in temperature increases metabolic function and increases healing. So the advantages are it makes it harder for bacteria to survive. We're going to find out through interferon that it makes it difficult for viruses, and it also speeds tissue repair. In addition, fever decreases the release of iron and zinc from the body, which means that these things, again, are not available for the pathogens to use. So you're kind of playing a game here where you're making it difficult for everybody, hoping that you as the host will outlive the pathogen, right? And of course, a fever, as we're going to find with most of these defenses we start talking about from now on, is not something that you want happening for a long period of time, right? Even a low-grade fever for a long period of time can be really damaging to the body. But we have to remember with these innate defenses, right? What was the goal? The goal of intact skin is to protect. The goal of these innate defenses is to protect if the skin has been damaged. And really, they're just there to hold the line until the immune system, who can come in and formulate an adaptive, specific response, can actually get in there and fix the problem. So fever was never designed to be a long-term solution. Fever is meant to be a short-term solution, which increases transferins, increases interleukin-1 activity, increases interferon, increases tissue repair, and decreases the release of iron and zinc. Now, the problem with fever is that it causes high blood rate, or excuse me, it causes high heart rate, tachycardia. It causes you to hyperventilate, which is tachypnea. Tachypnea, if you breathe really rapidly and shallowly, will cause blood acidosis. It can dehydrate you, and you can die from a high fever, right? A fever of 108, right, will cause brain damage, and a fever of 111 is fatal. So you got to be careful with fevers. A low-grade fever is good for a short period of time. High fevers are never good, right? So a fever over 102, you want to make sure you get that fever down, right? Because 108, you're talking about seizures and brain damage, and 111, you're talking about a person probably not surviving, especially young children. So that's defense number 2. So phagocytes, you can see, are working all by themselves, but here they're actually the ones starting this innate defense, right, by the release of those cytokines, which we saw in the previous slide. In addition to fever, we have a really interesting body defense that's called complement. And complement is kind of a funny defense. Its name is a little bit confusing, right? Because complement means to add to or to help with, right? And the reason that complement was called complement is because people noticed that there was something in the blood that could help the immune system to work. So the classical complement pathway, you can see there at number 1, there are these Y-shaped things, and those are actually antibodies, right, which are part of the immune system, which we haven't studied yet. But those antibodies have bound to and recognized a pathogen, and then that C1 protein is a complement protein, which recognizes those antibodies and then becomes activated. And when it becomes activated, the complement is just kind of this, it's sort of like a domino, right? You knock the first domino down, the rest of the dominoes fall, and it's just this cascade of proteins that were already present in the bloodstream activating. And that's the classical pathway of complement activation because it was the first one that was discovered. So the second that you see here in the middle is the alternative pathway of complement activation. And it's actually a more direct and innate thing, but it was not the first one discovered. So it was called an alternative to the classical pathway. And that is where the microorganism itself is directly recognized by a complement protein. And that complement protein causes this cascade of this domino effect of these proteins in order to activate the complement pathway. And then thirdly, we have this lectin pathway where little molecules that bind to the surface of the microorganism. This is especially important for binding to gram-negative bacteria. These lectins bind to the microorganism, and then the complement proteins recognize that the lectins have bound. But what's important to see here is whether you're talking about activation of complement because of the immune system, activation of complement all on its own, or activation of complement because of some innate factor, the result is going to be the same. And the result is inflammation, cytolysis, and opsonization. So inflammation, that same process that we talked about earlier, right, that causes the local dilation of arteries and the leakiness of local capillaries, that can start because tissue has been injured, and that can also start because complement has been activated. And it doesn't matter whether it's the classical, the alternative, or the lectin pathway, each of those three can result in inflammation. The other thing that each of those three can result in is cytolysis, right? So cytolysis, cell lysing, and the cell that's being lysed is the pathogen. And you can see a scanning electron micrograph just underneath that diagram of cytolysis. On the left-hand side, you see cells prior to complement activation, and then you can see just next to that cells after complement has been activated. Complement activation results in sort of a punching through of the cell wall, which causes the cell to rupture. And again, cytolysis, like inflammation, can be activated through the classical, the alternative, or the lectin pathway. And likewise, opsonization, the third result of complement activation, can also happen as a result of classical, alternative, or lectin activation. And opsonization, right, if you can kind of tell from this picture, there's kind of this weird like two-pronged thing coming up, and that's actually the end of a phagocyte, right? So that is the end of a phagocytic cell containing those PRRs, right? But for some reason it hasn't been able to recognize a PAMP on this cell, so it's been unable to phagocytize these cells. Well, what it can recognize is complement proteins bound to the surface of the cell. So those little red dots that you see on the surface of each of those 3 bacterial cells, those are complement proteins. And the white blood cell will actually bind to the complement protein and then engulf the bacterium and phagocytize it. And opsonization is the technical term for the enhancing of phagocytosis. So opsonization is an enhancement of phagocytosis. So you can see here, a complement, in addition to directly working to kill microorganisms, can also recruit innate defenses. It can recruit inflammation, which can bring white blood cells into the area, and then it can cause opsonization. And in that way, it's kind of interacting with the immune system as well, because once it brings those phagocytes into the area, what are the phagocytes going to do? They're going to phagocytize, they're going to themselves increase the innate defense, and they're going to communicate with the immune system, right? So that this complement process, this cascade of serum proteins, can be what it needs to be, and that is a very short-lived defense, because it can be very destructive having these complement proteins roaming around and causing cytolysis and inflammation and opsonization and all of that stuff. So complement needs to be very short-lived, which is totally fine, because the whole point of all of these defenses, right, is to just hold the line until the immune system can come on board. All right, so we've talked about the innate physical barriers, we've talked about phagocytosis, we've talked about fever, we've talked about complement, and now it's time to talk about the last of the innate defenses, and that is some interferons. Now, interferons are more specific than anything else we've talked about to this point, because interferons are targeted particularly toward viral infection. So the name interferon is actually meant to indicate to you that they're interfering with viral infection. Now, your book says that this happens in result to RNA viruses. That is not true. Interferon is a defense against any virus. And here's what kind of happens. So a virus, you'll remember from chapter 13, right? Step 1, attachment. Step 2, penetration. Step 3, biosynthesis. Step 4, maturation. And then step 5, release. And when step 5, release happens, that means everybody anywhere near this infected cell is in danger of being infected themselves. So interferon works to warn all of the other cells that there's a virus inside it. And this is kind of what that process looks like, right? So step 1, a virus from an infecting, a virus moves into the cell, right? So moves into the cell, the viral RNA or viral DNA is, and then, right, steps 1 and 2 are done. So now it's going to move on to step 3, right? It's going to start working to do its thing. But what it doesn't realize is that at the same time that the virus is undergoing biosynthesis, the cell is also transcribing and translating interferons. And those interferons are going to be transcribed and translated and released from the cell, not to do anything to help this cell in the least, but in order to send out a chemical warning that there's a virus replicating inside this cell. So the alpha and beta interferons, by the way, alpha, beta, it doesn't matter. Interferons are secreted by the cell, and then they're bound by a neighboring cell. And when the neighboring cell sees those interferons, it goes, uh-oh, right, there's viruses in the area. And so it kind of stops everything it's doing and starts transcribing and translating antiviral proteins. And once it does that, it's protected against the viruses that are quickly going to be in step 5, right? Because what was the whole point of the virus infecting the first cell, right? Attachment, penetration, biosynthesis, maturation, and release of the viral progeny, which will then go to this neighboring cell, but this cell knows they're coming, and so they blow up the virus when it gets into the cell. Now, sounds like a great strategy, right? Why doesn't that work all the time? Why do we ever get viral infections? Well, imagine that this cell, this neighboring cell, is a liver cell, and its job is to detoxify your blood or to make glucose for you, or maybe it's a pancreas cell and it's supposed to be making insulin. And instead of doing any of those things, it's just sitting there making antiviral proteins waiting to be attacked. Well, that's going to damage the body. So like complement, interferon has to be a very short-lived defense. And again, that's okay. That's what it's meant to be, because the idea is hold the line, protect the body, try to keep things under control while the immune system is working, right? Because The other thing that's going to happen, right, if you can picture in your mind, is when step 5 happens and all of these viruses are released, the white blood cells are going to come into the area, they're going to phagocytize the virus, and they're going to bring that virus to the lymph node, show it to the to the immune system, and the immune system is going to mount a specific targeted adaptive defense to that particular virus, right? So interferons are any virus. The immune system response will be targeted toward a particular virus, and because it's a targeted specific response, it doesn't have all of the problems that fever and complement and interferon have, where they're sort of hijacking large swaths of the body and actually, if left for long periods of time, become damaging themselves. So that's it for the innate defenses. As I mentioned, you have a slide in the lecture notes where you can see each of them summarized, and that's it for chapter 16. So that means you're welcome to go and check the chapter outline, check the study guide, check the animations at the end of the study guide, and then take that chapter 16 online quiz to make sure that you've got everything straight and you're ready to go for our exam.